Cells are the most basic units of organisms, the smallest unit capable of performing life functions and only observable under a microscope. The cell theory states that all living things are made up of cells and that all cells come from pre-existing cells. And that all comes into play during mitosis. Mitosis is asexual cell reproduction where one parent cell produces two daughter cells that are identical copies, making both daughter cells identical to the parent cell. Its purpose in unicellular organisms such as bacteria and plankton is reproduction. In multicellular organisms such as humans and animals, mitosis is a mechanism for growth, repair, development, and renewal. In the cell cycle, mitosis happens during the meiotic phase, which also includes cytokinesis. The phases before this are all under interphase. This is where the cell grows and gets ready to divide. Mitosis itself is the process where the cell divides. Mitosis occurs in 4 phases: prophase, metaphase, anaphase, and telophase. So after interphase, mitosis starts with prophase. During prophase, chromatin, a loose combination of DNA and proteins, coils into visible double-stranded chromosomes, long continuous thread of DNA that consists of numerous genes and genetic information, and forms sister chromatids, which are identical double-stranded chromosomes created before a cell divides. The nucleolus disappears, the nuclear membrane breaks down, the centrioles migrate to opposite sides and form meiotic spindles. Then the cell moves on to metaphase. The spindle fibers attach to the centromeres, which are regions where the sister chromatids are joined together and looks pinched. Double-stranded chromosomes are lined up at the equator or the midline of the cell by the spindle fibers. Each double-stranded chromosome consists of identical sister chromatids. Next, the cell goes through anaphase. During anaphase, sister chromatids separate from their partners and each chromatid is now considered a single-stranded chromosome. The chromosomes are then pulled to opposite poles of the cell. Finally, the cell goes through telophase, which can be considered the events of prophase reversed. With one chromosome at each pole of the cell, the spindle fibers disappear. Then the nuclear envelope will form around the daughter chromosomes. The chromosomes then uncoil and lengthen, forming chromatin, and the nucleolus reappears in each new nucleus. Mitosis ends with telophase, however the cell must go through cytokinesis to completely finish the cell cycle. Cytokinesis is where the cytoplasm divides and it happens immediately after telophase. In animal cells, the formation of a cleavage furrow separates the cytoplasm. In plant cells, the formation of a cell plate separates the cytoplasm.